Well, maybe Jim, while you're getting your slides up, I wanted to first take this opportunity to congratulate you and your team on 20 years of the Neurotech Leaders Forum. Really fantastic accomplishment. And
Thank you.
as well, from those of us out in the field, to thank you and your team for the tremendous service that you provide by gathering together all of this information that you provide to us in Neurotech reports. And I'm also excited to hear about the launch of bioelectrics reports and look forward to that as well.
Thank you, Warren. All right. So we just needed to get you onto the projector, which you now are. So go ahead.
All right. Thanks, Jim. Yeah, so before I get started today, I'm required to make some mandatory financial disclosures that are listed here. None of these really affect what I'll be speaking about today. So I'm going to go through 4 topics this afternoon in this organization of what I call realizing the potential of neurotechnology. First, a little bit about the promise of neurotechnology. Why is this such an exciting field? Second, and very briefly, a little bit about the principles of neurotechnology. What's the fundamental idea behind the technology? Through some products, and then spend most of my time here in what I call pitfalls, and giving you one very specific example of a pitfall and what can we learn from that so that we can move forward successfully with future opportunities. So first, we all understand quite well the huge challenge that we face with neurological diseases, disorders, and injuries, which are myriad and continuing to grow as a result of an aging population. And unfortunately, this is an area where pills will not save us. So shown on the left are some data from Science, a decade old, but still true today, which show first at the top the clinical development time for new drugs in the neuro area. And you see this is about 9 years. The clinical approval success rate, that is drugs that made it through the development pipeline, less than 10%. And then at the bottom, the cost of doing so, and this was $1 billion a decade ago, so it's probably 2 to 3 billion now. And this isn't just my view. If we look at some quotes, this is from Andrew Whitty, the former CEO of GSK, in a press release where they were disinvesting in these areas. Pain, depression, and anxiety are areas where we believe the probability of success is relatively low, and we think the cost of attaining success is disproportionately high. Similarly, from the former director of the National Institutes of Mental Health, and remember, the sole directive of the National Institutes of Mental Health is to treat mental health disorders. There are very few new molecular entities, very few novel ideas, almost nothing that gives any hope for the transformation of the treatment of mental illness. Really dismal statement from someone whose job it is to, in fact, find treatments for these challenges. But devices might. So the treatment of the diseases and disorders that affect the nervous system represent one of the most exciting areas for the life sciences, and medical devices represent one of the most elegant solutions for many of these problems. And this quote is probably 15 years old, but I think it holds true today as well. Why? Why are we in this position where devices might allow us to use electrical stimulation and recording to treat these challenging diseases and disorders of the nervous system? Well, first, we have solid fundamental underpinnings. And what I mean by that is that we are able to, we understand well how to deliver electrical stimulation without damaging the nervous system, and we understand well how to record from the nervous system. We have demonstrated safety, efficacy, and clinical utility and cost effectiveness of a range of devices, restoring hearing, treating chronic pain, restoring bladder function, treating movement disorders, treating epilepsy, etc. Thanks to the microelectronic industry, we have very reliable and very small implanted devices that allow us to do this. But it's important to recognize that, in particular, when we talk about functional restoration, we are still far short of normal nervous system function. So Jim quoted Paul Laviolette earlier. He's a popular guy today. And this is a quote from him again, quite some time ago. The neurostimulation market has the potential to be a mega market, similar to stents and heart rhythm management, with annual sales of $4 to $5 billion, fueled by growth in therapies to treat epilepsy, depression, chronic pain, migraine, headache, and obesity. And I think we're probably still a little short of this number, but rapidly approaching that number total. So that's some of the promise of neurotechnology. What about a little bit on the principles? What's the fundamental principle that underlies these devices? Well, I think about this as the process of information exchange with the nervous system. Electrical stimulation allows us to introduce information into the nervous system and exert external control. For example, allow someone to hear or cause a muscle to contract. And we can use that both as a therapy and for restoration of function. And then conversely, electrical recording is extracting information from the nervous system. We can use that to determine state. And importantly, this provides a command or feedback signal for a closed loop device. This information exchange occurs through a binary code of what are called action potentials. So these signals are either 0 or 1s, and it's the rate and pattern of these action potentials that either code information when we stimulate or code information when we record. So thinking about control, for example, of your bicep muscle under an able-bodied condition, you would think, hey, I want to contract my bicep muscle. That's going to generate a series of action potentials that travel down the nerve to the neuromuscular junction. Once they arrive at that neuromuscular junction, neurotransmitter is released. In the case of a skeletal muscle, this would be acetylcholine. And that causes some postsynaptic action. Again, in the case of your bicep muscle, it would be contraction of that muscle. But this could also be another neuron somewhere in a chain, and that could either be activation of that neuron or inhibition of the neuron. So this is what happens under able-bodied control. What about when we're doing this electrically? Well, fortunately, the same thing happens with electrical stimulation. You apply electrical stimulation and generate propagating action potentials. They propagate to the end, to the nerve terminal. Once they arrive, that nerve terminal has no way to know that those action potentials were generated by an electrode or by your brain. So they release the same transmitters in the same way, and they produce the same postsynaptic effect. So it's largely a trick. That is, you trick the nervous system because it can't recognize the difference between artificially generated and biologically generated action potentials. The advantages of doing this, in contrast to taking a pill, are that we can be quite selective in space and time. That is, only stimulating neurons that are close to an electrode, for example, and not stimulating those that are further away. And while those same neurons may share postsynaptic receptors, so that a drug will influence both postsynaptic targets, in the case of stimulation, we can be selective and only influence one of those postsynaptic targets. Similarly, we can readily grade electrical stimulation in real time. So in this example, if we turned up the intensity of stimulation, now we stimulate not just that neuron that's closest to our electrode, but the one that's further away, and influence both postsynaptic targets. Whereas in the case of pharmaceuticals, this grading is much more uncertain and much slower in time. So that's, I teach a course here at Duke on the fundamental principles of electrical stimulation. You just got the first 10 minutes of that class, and so come by and take the rest of it. Products. So this is a slide showing the range of approved in the green color and experimental in the black color applications of electrical stimulation for restoration of function. And you see this is myriad and growing every year. And remarkably, if we look at these approved devices, a huge number of them have come from academic research labs. So the largest part of our space is spinal cord stimulation for chronic pain. This was invented at Case Western Reserve University by neurosurgeon Norm Shealy and biomedical engineer Tom Mortimer. Cochlear implants across 3 of the manufacturers developed in academic research institutions, all the way up through retinal stimulation for blindness developed by Mark Hermione, first at Johns Hopkins and then at the University of Southern California. So it's important to recognize that the academic research lab is an area that can be mined for innovation and translation to develop the next generation of these products. And then I like this diagram from neurotech.com because it gives this idea of an ecosystem. And I think it emphasizes that we have this fundamental research and development going on at universities and non-for-profit research institutes. Then we have investors who are taking these innovations and translating them, creating companies, and those companies are living outside on this outer ring of the circle. So that's a little bit about products in this space. And then finally, pitfalls, where I'll spend most of my time this morning. So we're subject to pitfalls because there are a large number of challenges to being successful in the neurotech space. We have long and expensive product development cycles, long, risky, and expensive product approval cycles, very crowded space of existing intellectual property that can be challenging to navigate if you're a new player in this area. Every element of what we do is highly regulated, from the development through the manufacturing, through the marketing, through the sales, through post-approval studies, et cetera. In contrast to, for example, a simple prescription that you might get from your cardiologist, deployment of these devices often requires a complex team of trained specialists. So it's pretty unusual that you only will be dealing with a single physician or a single healthcare professional to deploy a neurotech device. And of course, these devices are relatively expensive and dependent on third-party reimbursement for revenue. So if we think about time, most of fundamental R&D is occurring up here in the inside part of that circle that I showed you. Maybe some of the early clinical feasibility is also done in that inside circle. Then translation through startup or acquisition to pivotal multi-center clinical trials, filing of a PMA, and eventual approval of that PMA. So after the fundamental R&D is completed and you enter this pipeline, it's still a 5 to 8 year endeavor. And so what I'm going to focus on today is really this question, which should be answered at the onset of entering this pipeline. But in the examples that I'll show you, I think, at least in my opinion, really insufficient heed paid, if you will, to paying attention to what are you stimulating and how does it engage your therapeutic mechanism. So I'm going to focus on a particular application of vagus nerve stimulation to treat heart failure. So as many of you know, vagus nerve stimulation has been used both successfully and unsuccessfully to treat a large range of disease and disorders, from its earliest use in epilepsy, its most recent use in targeted plasticity for stroke restoration, and along the way, it's also been used as a treatment for chronic heart failure. And this began with some preclinical studies in a rat model. And in this rat model, if you administer a myocardial infarction or a heart attack, you see that the animals, after about 3 weeks, begin to die. And by about, what would this be, about 6 months out, about half those animals are dead as a result of heart failure occurring as a result of that initial myocardial infarction. So what was done in this animal model was to deliver electrical stimulation of the vagus nerve, and the amplitude of stimulation, and this is quite important, was titrated to produce a 10% reduction in heart rate, or what we would call bradycardia. So you turned up the intensity of stimulation until the heart rate declined by 10%. That was the dose. And the effect of that therapy was quite dramatic in this animal model. So you get an initial loss of about 10% of the animals. This is occurring before treatment is even delivered. But in those animals that receive treatment, you see that the vast majority of them are still alive at the end of the study, in contrast to the animals who did not receive vagus nerve stimulation. So a 70% reduction in the relative risk of death, pretty dramatic outcome. So this rat study was followed up with subsequent studies in dogs. And again, electrical stimulation was applied to the vagus nerve at a dose that reduced the heart rate by 10%. This produced significant improvements in cardiac function. This shows you the left ventricular end diastolic volume. So how much blood is left in the ventricle after it contracts? You see that in the control animals, there's a substantial amount of blood left in the ventricle, whereas that's significantly and substantially reduced by vagus nerve stimulation. Same at the end systolic volume, and thus the ejection fraction is significantly and substantially increased by vagus nerve stimulation. The effect of vagus nerve stimulation exceeded what's achieved with traditional medical therapy with beta blockers. So this shows you the left ventricular ejection fraction before and after therapy in the control group. In a group of animals that received beta blockers, you see a significant and important improvement in ejection fraction. But in those animals that received beta blockers plus vagus nerve stimulation, you see a superadditive effect. That is, vagus nerve stimulation increased even further the improvement in left ventricular ejection fraction. So extremely promising preclinical data in 2 different animal models. This led to a clinical feasibility study in human. And this study enrolled just over 30 patients, and these patients were quite sick. So 15 of them, and I'll tell you what these classes are in just a second, in class 2, 15 in class 3, and 2 in class 4, and left ventricular ejection fractions less than 25%. So these individuals are quite sick. For those of you who are not cardiologists, the New York Heart Association has these classes where class 1 is cardiac disease but no symptoms, and class 4 is people experiencing symptoms even at rest. So these patients basically can't get up and move. Most of the patients in this study were in class 2 and class 3. So shortness of breath as you got up, went up the stairs for something, and marked limitation in activity due to symptoms. So unable to walk beyond very short distances. Similar to the preclinical studies that I told you about, electrical stimulation was delivered to the vagus nerve, and it was titrated to produce a 10% reduction in heart rate. So this shows you the heart rate as a function of time recorded in one of these patients. And the blue is when stimulation was turned on, and the red is when stimulation was off. So you see every time stimulation was on, the heart rate declines. Stimulation goes off, the heart rate goes back up. Stimulation goes on, the heart rate declines. Stimulation off, heart rate goes back up, etc. So this is how they determined the dose or how much stimulation was being delivered, comparable to what we saw in the preclinical animal studies. And it works. So this shows you changes in left ventricular ejection fraction at baseline after 3 months of therapy, and that effect was preserved at 6 months of therapy. So a substantial and significant improvement in left ventricular ejection fraction as a result of vagus nerve stimulation. There's no control group here, so the patients act as their own control, and you're just comparing the patients at 6 months to the patients at baseline. This shows you the change in that New York Heart Association class. Well, remember, 4 were the sickest patients, 3 were next sickest, and there were none who were not very sick in class 1. 3 patients died, not unexpected in a trial like this, where patients are quite sick. But notice that the vast majority of participants in the study got better by at least 1 full class. A couple got worse. And that this process of improvement either was maintained or improved between 3 and 6 months. So you can think about this as like a flip-flop between lots and lots of sick patients to fewer and fewer sick patients as a result of this vagal nerve stimulation therapy. So it's always important when we talk about these kinds of studies to consider not just efficacy, but risk. So as I told you, there were 3 deaths apparently unrelated to the therapy. Again, these patients are quite sick. 26 serious adverse events, which affected 41% of the participants. One had a surgically related pulmonary edema, pretty significant adverse event, and 1 loose electrode connector requiring reoperation. As I teach my students, this is the fault of the engineer who designed this electrode connector, because it should have been designed in such a way that no matter how distracted the surgeon was, they couldn't have a loose electrode connector. There was pain at the stimulation site, and then typical side effects that result from vagus nerve stimulation: cough, disruption of voice, and pain in the jaw or neck. So kind of unremarkable side effect and adverse event profile. So these studies, as you might imagine, led to a huge amount of interest in vagus nerve for the treatment of low ejection fraction heart failure, and a number of companies then subsequently launched pivotal clinical trials based upon these data that I've described thus far. However, those clinical trials failed. The first reporting out was Boston Scientific faced a stinging setback for vagus nerve stimulation device in mid-stage trial. Vagal nerve stimulation, as delivered in the Nectar heart failure trial, failed to demonstrate a significant effect on primary and secondary endpoint measures for cardiac remodeling and functional capacity in symptomatic heart failure patients. Boom. Another one. Results from Innovate heart failure trial indicate that vagus nerve stimulation does not reduce the death rate or heart failure events in chronic heart failure patients. What happened? Right? The trials failed, and we spent a lot of money on these trials. What went wrong, given the promise from the preclinical studies and the clinical feasibility study that I described? Well, the hypothesis that I'm going to explore and hope convince you of is that the dose that was delivered in these trials was wrong. And the reason it was wrong is a result of therapy-limiting side effects. These are data from the initial or one of the pivotal clinical trials of vagus nerve stimulation for treatment of epilepsy. And the way the dose was determined was on initiation, that is when you begin stimulation, the current was increased from 0 to a level perceived by the patient yet tolerated. So that is, turn it up until the patient doesn't want you to turn it up any higher. And that produces, as you might expect, and we've already discussed, changes in voice, because the vagus nerve innervates the muscles of the skeletal muscles of the airway and throat, cough, and pharyngitis, or sore throat. These are examples of similar quotes, but now in cases of vagus nerve stimulation for heart failure. During therapy titration, the stimulation amplitude was increased until patients experienced side effects that were unpleasant, neck pain or coughing. And in another trial, common device-related adverse events after VNS titration were transient mild dysphonia, disruption of speech, cough, and oropharyngeal pain. Same kinds of things that were described in vagus nerve stimulation for epilepsy. So if we want to understand what's the cause of these side effects, we need to be aware that the vagus nerve, this shows you a cross-section through the vagus nerve from a human, is composed of a very heterogeneous population of nerve fibers. This shows you an electron micrograph zoomed in on one of these collections of nerve fibers. You see there are these very large diameter myelinated axons. These might be 10, 15 microns in diameter. Small diameter myelinated axons, and then a lot of very small 1 to 2 micron diameter unmyelinated axons. And these different axons have different functions. So we wanted to understand, how do the complement of nerve fibers that are activated depend on the stimulation parameters that are applied to the vagus nerve, and how do those complement of nerve fibers that are activated influence the therapeutic effects of vagus nerve stimulation? So in order to do that, and these experiments were led by Paul Yu, who was a postdoc in the lab at the time and is now a professor at the University of Toronto, we implanted vagus nerve stimulation system on the vagus nerve of anesthetized dogs, where we could deliver stimulation and then downstream record from that same nerve. This shows you an example of that recording called the compound nerve action potential as a function of time across a range of stimulation amplitudes. And this first component of the response is the stimulation artifact as a result of applying the stimulation pulse. And that's then followed by activity from the nerve fibers that you stimulated here, and the action potentials are traveling down the nerve, and you can record from that nerve down here. And notice that as we turn up the intensity of stimulation, both the amplitude of this response grows and we recruit additional peaks. And these additional peaks are reflective of activation of these different types or different populations of nerve fibers within this heterogeneous nerve. So we measured how much current did you have to apply, what was the threshold amplitude to stimulate the largest diameter fibers. Those fibers actually are those that innervate the skeletal muscle of the airway and throat and produce the side effects. Intermediate size myelinated nerve fibers, these are sensory fibers carrying information from the organs of the thorax and abdomen up to the brain. Small diameter myelinated axons, the B fibers, those would be these fibers here, and the unmyelinated C fibers. So this tells you how much current you had to apply to activate these different populations, but what were the physiological effects of these different kinds of nerve fibers? In order to establish that, we measured the changes in heart rate that occurred as a function of the stimulation intensity. And this is an example of an electrocardiogram. So each spike is a single heartbeat, and you see that the heartbeat gets further apart. The heart has slowed down, or bradycardia, during the delivery of stimulation. And then after stimulation ceases, the heart rate immediately recovers. So we could also measure the threshold to produce this reduction in heart rate. And that's shown here. So this shows you the stimulation threshold required to produce activation of the largest myelinated axons, that is, those required to produce side effect, and the amplitude that you had to deliver to activate these small diameter axons. They're the ones that produced the reduction in heart rate, or bradycardia. And that required a 9-fold increase in intensity, from about 0.5 milliamp all the way up to about 4.5 milliamps. And remember, those preclinical studies and the clinical feasibility study all used intensities of stimulation that produced bradycardia, or that 10% reduction in heart rate. So what about the pivotal clinical trials? What intensities did they use? Here's one of them. The self-reported threshold for activating laryngeal fibers, that is, these largest diameter axons, was about 1 milliamp. And by they were able to turn that up only to about 1.2 to 1.4 milliamps. That's the maximum that the patient could tolerate. So instead of increasing by 9-fold, they only increased the intensity of stimulation by 1.2-fold. In other words, they successfully activated the axons in the nerve that produce side effect, but highly unlikely that they produced any activation of the axons that produced bradycardia, and thus the desired clinical effect on heart failure. Here's another trial. At the end of the 6-month follow-up period, the average output current was 2.2 milliamps. This was only a 1.7% increase in amplitude from that was required to produce side effect. And no patients were programmed to a VNS intensity that elicited side effects or an acute heart rate reduction of 4 to 6 beats per minute. So they were specifically avoiding reducing the heart rate, even though the preclinical and clinical feasibility studies were all done at intensities that produced a 10% reduction in heart rate. And then finally, the third failed trial went from 1.4 at least these were the first two, 1.4, 2.0. This one turned it up all the way to 3.9 milliamps. But again, no difference between control and vagus nerve stimulation on heart failure related events. And their conclusion, although higher currents were achieved, this hypothesis was not supported by our study, as the primary outcome did not improve with vagus nerve stimulation. And they only used a 3.3-fold increase in amplitude, quite a bit less than the 8-fold increase that we established in the preclinical studies. So, what are you stimulating and how does it engage your therapeutic mechanism? This is an example where the pivotal multi-center clinical trials were not done in such a way as to adequately engage the therapeutic mechanism. That is, they did not stimulate those small diameter B fibers that were required to produce a 10% reduction in heart rate, and in the fundamental and clinical feasibility studies had shown that that was sufficient to treat the symptoms of heart failure. Just want to give a little bit of preview that this problem, in my opinion, is only going to get worse. And the reason it's going to get worse, use the following quote to motivate this. Rising adoption of neurostimulation devices by physicians and the increasing prevalence of chronic neuromuscular disorders and others, of course, are major factors predicted to drive the market for implanted neurostimulators during the forecast period. However, the discomfort caused by invasive surgical procedures for implanting the device, which is also expensive, is a major restraint for the market. And they're talking here about the move from surgically implanted devices to less invasive or minimally invasive or non-invasive devices. So there's this push to non or minimally invasive devices. And I show a couple of those here. Here, stimulating the auricular branch of the vagus nerve in the ear, ostensibly. Here, ostensibly with a surface stimulator stimulating the vagus nerve as it courses through the neck. But very little understanding of what's actually being stimulated and how whatever they are stimulating in these cases engages the relevant therapeutic mechanism. So to recap, I think to realize this great potential of neurotechnology, which I highlighted through these promises, we need to pay heed to the principles of stimulation and what's operating. This is going to give us tremendous opportunity to develop new products, right? There are lots of activity in the middle of that circle that can readily be translated, but we need to beware of these potential pitfalls, and in particular, what neural elements are being stimulated and how that engages the relevant therapeutic mechanisms. I'd like to acknowledge contributions to the work that I described today, both from Paul Yu and from Juan Hincapilla, who was a scientist at Boston Scientific at the time, who sponsored some of that work. And then as well, contributions from Nikki Pillow and Eric Musselman, and financial support from the National Institutes of Health on our current efforts to understand what is being stimulated by vagus nerve stimulation and how that can be controlled through appropriate design of electrodes and applications of appropriate stimulation parameters. Thank you very much for your attention, and I'm happy to take any questions. Can't hear you on Zoom, Jim, if you're speaking right now.
Okay. Can people hear me now?
Yes.
Okay, super. Button, turn your laptop audio off. That feedback's coming from the call was coming from in the house. All right. Thank you, Warren. That was a great talk. Is there anyone on Zoom? I want to give Zoom people a first crack at a question. See, anyone raising their hand? Baden? All right, Michelle, do you want to? I'm sorry?
Yes. Thank you.
Go ahead and unmute yourself.
Thank you for the talk. Do you believe that there is an electrode designed for the cervical vagus nerve in humans, which can selectively activate B fibers, and what that might look like?
Okay, Warren, did you hear that?
Yeah, I did. And I'll just repeat it for make sure the audience heard it as well. So the question is it possible to design an electrode for the vagus nerve that can activate selectively the C fibers? And I presume you're implying
Sorry, the B fibers for the heart rate trial.
The B fibers. Yeah, so there have been efforts to stimulate selectively these smaller diameter nerve fibers. So let me just go back here and I can use this cartoon. I'll use this figure here, would work fine. And the idea is to stimulate everything and then block the large fibers further downstream. And there have been a couple of methods developed to do this over the years, and they can be demonstrated in vivo in a laboratory setting, but none of them are particularly robust, meaning they require a lot of fiddling to get them to work. And so we need methods for clinical deployment that require no fiddling at all. So I think it's still an open challenge to develop really robust methods that would allow you to only produce action potentials in the small diameter myelinated axons without getting simultaneous activation of the large diameter side effect producing axons.
Okay, Warren, I'd like to ask the next question. And that has to do with our 20 years covering the neurotech industry. You and I met 20 years ago, roughly. And you've been on the academic side, first at Case Western Reserve and now at Duke. You've also been on the commercial side of a number of startups. So you've had a unique opportunity over those 20 years to see a lot of successes, a lot of failures, a lot of scientific advances, a lot of engineering advances, a lot of commercial successes. I wonder if you would be willing to give us your impression of the most significant scientific and the most significant commercial advances that you've seen over the last 20 years in the field of neurotechnology. Don't want to put you on the spot, Warren, but
no, I think
give us your thoughts.
Put on the spot, when pressed for an immediate answer, I think scientific is for me relatively easy, and that is the advent of optogenetics. And for those who don't know, optogenetics is a way that we can render neurons susceptible to stimulation by light. And this can be done in such a way that we can use genetic targeting approaches to render only very select populations susceptible to light. So that is, even if you put an electrode into a very heterogeneous population of neurons, you can, by only having certain of them rendered susceptible to light, stimulate selectively only those neurons. This, in my opinion, is the most important advance in neuroscience in the last 20 years. There are some very early clinical applications of this, in particular to restore the sensation of sight. Whether it will result in clinical neurotechnologies, I don't know, but on the science side, that's the most important advance.
Yeah, and if I can interrupt, Warren,
yeah.
You may have heard that just recently, Karl Dieseroff and colleagues were awarded the Lasker Prize.
Yep.
The Lasker Prize being a very significant a prize, just probably below the Nobel Prize. But I also wanted to interject that one of the first, if not the first, articles in a industry publication on the subject of optogenetics was written by none other than Warren Grill in Neurotech Business Report. So I want to make sure you get credit for that, Warren. But yeah, go ahead and give us your thoughts on the most significant commercial product.
I think on the commercial side, the most important advance is closed loop brain stimulation. And I think the most obvious example of this is the responsive neurostimulation system for the treatment of epilepsy. And what I think is intriguing about that is not only is this an effective therapy, but it's also a tool that is allowing us to learn a lot more about epilepsy. So for example, this chronic ongoing recording of brain activity being correlated with seizure allowed people to determine that there's great periodicity to the probability that seizures will occur within a day, within a week, within a month. And even in the absence of a therapeutic intervention, that information can be extremely helpful to an individual with epilepsy. That is, what is the probability at any given time that I may have a seizure? We're also seeing now early examples of closed-loop deep brain stimulation for the treatment of Parkinson's disease, for the treatment of essential tremor. A recent example of brain stimulation, closed-loop brain stimulation for the treatment of depression out of Eddie Chang's group. And so to me, that's on the kind of clinical side, that's something that is really exciting and I think will spread beyond brain stimulation to other applications of stimulation as well.
Great. Okay, do we have any other questions for Warren on Zoom? Anyone with their hand raised? Alan, do you want to go ahead and unmute yourself? Can we hear Alan? Do you need to? On the speaker? We can't hear Alan.
I'll just read the question, Jim.
Go ahead.
Looking forward to the next 5 to 10 years relative to the last 2 decades, can you calibrate your expectations as to the pace of innovation in the sector? Yeah, I'm always a little bit reluctant to try to foretell the future. You know, there are two kinds of people who predict the future: fools and politicians, and sometimes the politicians are right. But I think for the reasons that I spoke about, that is this very strong foundation of success. We are really poised to undergo an accelerated growth in this area. And while I don't want to leave the impression that minimally invasive or non-invasive stimulation doesn't hold promise, because that's where I think a lot of this growth may actually occur. But I think we need to be cautious as a field that we don't allow that growth to outstrip our understanding of what those therapies are doing. Because if we do, we end up doing, replaying something like the North Star experience that Jim relayed earlier, where a big failure, is not isolated to that particular therapy or that particular company, but tends to put a stain across the whole field. And that's really where this idea of take your time, we're in a hurry comes from, which is we can't rush through this because these are important problems. We need new therapies. We're in a hurry to get those therapies. But if we try to rush through these important questions at the beginning, we're ultimately not going to be successful. And that's going to deprive people down the line of having these therapies available.
Okay, thanks, Warren. Anyone here in the room who has a question for Warren? Not seeing anyone else on Zoom. Okay, well, with that, Warren, we'd like to say thank you very much for your talk. It was a great talk. And again, thank you very much for the important role that you played with Neurotech reports. We certainly appreciate it. We're looking forward to the next 20 years to see what new commercial and scientific developments emerge. We know you'll have a hand in many of those. So again, thank you very much, Warren.
Thank you, Jim.